Okay, so I just first want to thank the organizers of this meeting for giving me the opportunity to talk here today. So I'm
You're not
very
sharing
excited.
the screen yet. So.
Oh, all right. That would help, right? And let me get back to my screen. Share, and there we go. I think that might be better.
Great, yes, we can see
Good.
it now.
Okay, great. Okay, so today I'm going to talk a little bit about some of the work that I've been doing the last few years at Southern Illinois University. I started my lab in 2017, so some of this data is a little bit on the newer side, and there's certainly a lot of different directions that we can go with some of these studies. But one of the projects in my lab is trying to explore the use of DPPC liposomes to capture polychlorinated biphenyls, or PCBs, which is a class of emerging environmental pollutants. So one of the overarching goals in my group is to try to develop and look at different types of biomaterials for different applications. So some of those applications include how can some of these materials, and what I'm going to talk about today is primarily lipid-based materials. So how can we use them to enhance the physical and chemical properties of things like different surfaces using films or coatings, environmental remediation, therapeutic applications, and if anybody's familiar with the liposome field, they've certainly been used very successfully in that particular area. Probably less notably have they been used in remediation and things like agriculture. So that's kind of an area that I'm trying to explore a little bit more with these liposomes. And when we study different biomaterials, one of the criterion that is important to us is that they are biodegradable, so environmentally friendly, and that when they break down, eventually they can be assimilated back into the environment without posing more of a problem than what was there originally. So we wanted to kind of explore a different class of molecules that could be incorporated into liposomes, because certainly there's a lot of interest and a lot of study in using liposomes therapeutically. So to see, where we could sort of find a really good problem to study and hopefully try to use these liposomes to help solve is, are there compounds in the environment that pose a threat? And certainly there are a bunch of them. So there's a class of persistent organic pollutants. If you look on the EPA's website, they've got a list of, let me see here, I'm losing my mouse. They have a list of about 12 that they call the dirty dozen. So I'm going to focus primarily on PCBs, or polychlorinated biphenyls, because that's kind of where we're starting out as far as looking at how we can use liposomes to capture them. And the issue with PCBs and a lot of these persistent organic pollutants is that they don't necessarily, it's not necessarily the quantities with which they're introduced into the environment, but it's the fact that they're not readily broken down. And so over time, they begin to accumulate. They can accumulate in soil, they accumulate in water, and then eventually they work their way into the ecosystem and different organisms. And so they can be really, really difficult to get rid of, and they can pose health problems potentially. So PCBs in particular saw their first use in industrial applications. So they were used in a lot of industrial equipment like electrical transformers. They've got very desirable cooling properties. So they were used as industrial lubricants, and they were also introduced as paint additives as well. And in general, polychlorinated biphenyls look something like this. They have a biphenyl ring structure that's attached, and then there are different sites of substitution for chlorine. So depending on the level of substitution, you can have different types of congeners. So it's usually when PCBs are synthesized, they're usually kind of a heterogeneous mix of these different congeners. So PCBs were introduced in the late 1920s, early 1930s, and they were officially banned in the late 1970s after concerns about their toxicity began to surface. So it became primarily a concern with people who were working directly with these compounds, so in manufacturing and things like that. And so the link was established. They were, potentially causing health problems, and so they were officially banned. So the issue is usually improper disposal of a lot of these pieces of equipment, electronic equipment, so that can kind of directly introduce this compound into the environment. So it can become a problem that way. And this compound was manufactured under different trade names depending on what part of the world they're being produced. So in the US, it's manufactured under the name Arachlor. And so in this particular study, we chose to look at Arachlor 1260, which is a specific congener, and it's substituted at the 2,2', 3,3', 4,4' position of the biphenyl system. So we're going to call this hexachlorobiphenyl, or HCBP, moving forward. So this is considered a heavy congener of Arachlor by virtue of the extent to which it's substituted. So if you kind of dig in the literature a little bit, what you'll find is this compound is incredibly insoluble in water. So really between about 2.5 to about 25 micrograms per liter is about all that would be naturally soluble in water. So very, very low water solubility. So it seemed like, just by virtue of the principle of like dissolves like, this seemed like a good candidate to try to work with and incorporate into our DPPC liposomes. So if anyone's familiar with liposomes or has worked with them before, they're very interesting in and of themselves. My background is membrane protein biochemistry, so we work with different lipid systems quite a bit, and that was kind of my first introduction into liposomes. And what I find pretty fascinating about them is that just by virtue of how they're prepared and handled, they can really assume very different physical properties. So one of the first questions we wanted to kind of decide from a reproducibility standpoint was how should we prepare these liposomes? What's the best way to do it? Because if there are any alterations in the way that they're prepared, this can really affect their physical properties. So liposomes can vary in size anywhere from micrometers down to 20 nanometers. And again, depending on how they're prepared, you can get small unilamellar, this is called an SUV, or small unilamellar vesicle, which is just another name for liposome. If you use things like extrusion, you can get actually very defined large unilamellar vesicles with a very defined size, and they're actually pretty homogeneous as well. So you're talking kind of the middle of the size scale. And then you can also use other methods to produce multilamellar vesicles as well. And these tend to be a little bit larger, and they're kind of on the micrometer scale. So we were looking for a very robust way to produce or to prepare these liposomes sort of in a batch setting. We wanted to kind of just throw these into a vial and kind of find a very straightforward way to produce them. There's certainly a lot of other methods that can be used. You could use dialysis. These tend to take a little bit more time, so we decided to use sonication because it tends to be very straightforward, and we can prepare a batch of liposomes in a matter of minutes. And of course, you can tune the properties of liposomes depending on the lipids that you use to prepare them. So I just have a bunch of different phospholipids listed here, but we decided to stick with a very simple, straightforward system just for proof of concept initially. So our liposomes are prepared with 100% DPPC, but certainly you can incorporate other things into them to alter their properties, either their surface charge, and also you can incorporate small molecules that can actually enhance their stability as well. So we wanted to see if we could capture PCBs using these pure DPPC liposomes. And we decided to use two approaches. One was the direct incorporation, where we would add our compound during the liposome preparation process. So that's what we call our direct incorporation, because presumably when you prepare them together, you get your HCBP compound incorporated into your liposomes. And we also decided to explore passive incorporation as well, where we added preformed pure DPPC liposomes to our compound that was dried on the inside of a vial, because I guess implementation-wise, it seems like it would be a little bit more straightforward if we had our liposomes that we just kind of added to a sample, if we're trying to, kind of use it for remediation purposes. So we prepared initially 5 different samples. Of course, we have our 0% sample where 0% by mole of HCBP is added to our DPPC liposomes, and when we went up to about 20% because we didn't necessarily want to overwhelm the liposomes with this compound. So we kind of tried to be somewhat conservative. So our total lipid quantity is about 25 milligrams, and of course that varies depending on how much HCBP is present. So ultimately we co-dissolve. Let's see. Lost my mouse again. We co-dissolve the compound and the lipid together in a vial out of using hexanes, which tends to evaporate quite easily from these 2 compounds. And then what we end up with, excuse me while I find my mouse, here we go. What we end up with is a solution that looks something like this. So initially, when we have our lipid and our compound and we start to sonicate, it's kind of a very thick, milky white suspension. And then over time, after we put it through several rounds of sonication, and we had to be very, very tight about this so that so that our preparations were as reproducible as possible. But eventually what we end up with is kind of this translucent looking solution. And if you go from left to right here, this is increasing concentration of HCBP. So qualitatively, you can kind of see that it's a little bit more opaque as we start to add our compound. And after we prepare our compound, we centrifuge to remove any particulates and any unincorporated lipid. And what we find is that as we increase a concentration of HCBP, we start to see this noticeable white pellet that kind of appears when we spin down the particulates after preparing our liposomes. So that was kind of an interesting observation there. So we used 2 methods to assess incorporation. We used just straightforward UV vis spectroscopy because it turns out Aurochlor 1260 actually has some very nice spectroscopic properties. So we can kind of use that to track where it's going and if we're actually incorporating it into our liposomes. And then we also used differential scanning calorimetry to assess the thermal stability of liposomes prepared with HCBP. So we can look at the temperature phase transition with and without HCBP for both methods of incorporation and see if we're seeing any appreciable changes in our liposome suspensions. So ultimately, with our UV vis measurements, there's kind of a lot going on this table, but if you kind of look on the left-hand side of this table, this kind of shows the direct incorporation. What we end up with is we can incorporate up to about 0.7 milligrams per milliliter. So if we compare that to our theoretical HCBP, we're getting about 28, or approximately 29% incorporation. And what's interesting is at a 1% mole quantity of HCBP in our liposomes, we can actually incorporate more. So what I think is going on here is that as we try to incorporate more HCBP, we're kind of compromising the ability to form liposomes in the first place. And that's why you kind of see this this decrease in the percent incorporated. Although ultimately, if you look at the quantities, we're able to incorporate more HCBP when we attempt to do it directly. If we look at passive absorption over here on the right-hand side of the column or right-hand side of the table, here we exposed our preformed liposomes to our HCBP film, which was dried on the inside of a vial, and we just basically let it incubate at room temperature. So we incubate at a minimum of 4 hours, and we went up to about 24 hours just to see if there would be a partitioning of the compound into our liposomes. And we used a buffer, just buffer without any liposomes present as our control. And what we found is after 24 hours, there was a negligible amount of our HCBP that leached into the buffer, which isn't completely surprising if you consider its extremely low solubility. But after 4 hours, we already start to see some incorporation of HCBP into our liposomes up to 24 hours. So, and this is from a 1 milligram total of material. So we get about 23% incorporation, and the rest is just stays dried on the inside of the vial. So so that was kind of exciting that we sort of had this very passive way of just capturing this compound from the vial. And certainly we can attempt to optimize this by increasing our lipid concentration to see if we can maybe increase those numbers a little bit too. So from our differential scanning calorimetry results, we didn't really notice any appreciable differences in our passive incorporation of HCBP, meaning that when we compared the thermogram to our 0% HCBP, there really was no appreciable difference in the transition melting temperature. So if you're not familiar looking at a DSC thermogram, what we're looking for here is you have a sample and a control that you heat up together to a temperature upwards of about 100 degrees C. And so you're effectively melting or kind of destroying your sample. If you're looking at a protein, usually you're surpassing the point at which it denatures, and that has a very characteristic TM, or melting transition temperature. So we decided to look at that for our DPPC liposomes. And it turns out that at 0% HCBP, we get a TM of about 40.8, which is consistent with what's reported in the literature. And as we increase our HCBP concentration, we start to see this transition melting temperature drop a little bit. So that kind of suggests that the incorporation of HCBP is kind of has a destabilizing effect on the liposomes. If we look at the thermograms, you can kind of see that with the incorporation of HCBP, we sort of see this peak broadening. And there's also this pre-peak here, which again is consistent with what's been previously reported as well. So we attribute this pre-peak as kind of unincorporated lipid that we weren't able to remove by centrifuging. But ultimately, we see this broadening. What's unclear from this data is if the heterogeneity, or the whether or not this compound is being incorporated into all liposomes equally. How homogeneous are these samples? I suspect there's probably a population of DPPC liposomes that have no compound incorporated, and then there's probably a population in the same suspension that has quite a bit incorporated. So that's something that we want to explore a little bit further, because we can't really differentiate that just from the DSC data alone. And so in summary, we can incorporate HCBP, which is a polychlorinated biphenyl. We can incorporate it into liposomes both passively and directly. Direct incorporation gives us higher yields, but it lowers the thermal stability of our liposome suspensions. So in terms of implementation, we might want to explore liposomes that maybe we can incorporate other molecules that might help to increase the stability of liposomes. What we'd like to do next is try to use this proof of concept on real soil and water samples to see how a different matrix, these were under very controlled conditions. So we'd like to see how using a different matrix would see if we can extract PCBs from a different type of matrix into liposomes. Also, optimizing liposome stability is another area of interest, and establishing some cutoff sensitivities to find out exactly, if we're below a certain threshold, will we be able to still use these liposomes effectively to extract this compound? And lastly, I kind of threw this slide in because, once we capture this compound into liposomes, what do we do next? We don't want to put it back into the environment. So where can we go from there? And that's where I think exploring some microbial-based solutions would be a possibility. So there was a nice review just put out this year that describes a whole host of aerobic and anaerobic organisms that are actually capable of degrading this biphenyl compound into benzoic acid and hydroxypentadienoic acid, which are far less toxic than the PCBs. So with that, I would like to, I'd like to thank my group. I've had a lot of students kind of come through my lab, so I'd like to thank them for their interest and thank you for participating. And I look forward to answering your questions.
Thank you so much. That was a really fascinating talk. When you're moving into soil and water samples, do you have a target you're going after? Or sort of a real world example that you think might be more approachable?
I think initially we would still probably explore very kind of do this under very controlled conditions and kind of, collect soil and maybe, kind of artificially add different quantities of the compound just to see if the liposomes can even capture it from this matrix. And if that looks promising, then we would have to kind of find areas of contamination and see what we can do.
Awesome. Yeah, this is a really exciting idea and it looks really promising. Thank you so much for sharing it with us.
Thank you.